Um, okay, to get it started, let's give you an outline of the presentation today. We are going to start with some background and importance of the research. Then we're going to move on to crop stress phenotyping strategies. Then I'm going to present you two case studies that I developed with my research in climate smart agriculture. The first one in water deficit priming, and the second one in seed priming. And then I come up with some concluding remarks. So to get it started, I always like bringing like some pictures of images around the globe of how lately we are seeing more climate extremes, we are having more limited water resources, and just highlighting that this is not an isolated issue. This is a problem happening everywhere and it needs our attention right now to improve like how we produce crops. Like we need to be prepared for a future that is going to be even more challenging, right? Because we're seeing more often climate extremes. We have an exponentially growing population. The trends are like just to increase the competition for natural resources. And agriculture already used like 70% of the water resources around the globe. And that only covers 20% of the land that is irrigated. Still, the majority of the crop production is done under rain-fed, not irrigated systems. And with more and more water scarcity or erratic rainfalls, we are going to start moving more towards like irrigated production. And where is that our water coming from, right? So that's why like I urge you to start thinking like how we can produce crops more efficiently in the future. Current like strategies to improve like water use efficiency or like how to improve like how we produce crops and valuing like our natural resources. We can manage the crops. We can improve our cultivars to make them more stress tolerant, right? Like this is like to some extent we can manage the plants since we cannot manage completely the weather. We cannot improve like rainfall frequency. That's one strategy through crop breeding. We also can manage to some extent our crop environment. We can use strategies such as regulated deficit irrigation, in which we strategically choose periods in the crop cycle to reduce the irrigation to improve the irrigation efficiency. We can use that as well to improve the water use efficiency of our crop production. And lately, crop physiologists have been manipulating crop physiological processes in the field with the goal of improving stress acclimation. And this would be using like the two previously mentioned strategies that I said to benefit in the process of improving stress responses. And in this case, we study genotypic material to understand genotypic differences to strategies like as regulated deficit irrigation, right? Here we are not trying to only improve irrigation efficiency, we are trying to also understand how the crop responds to that. And among the manipulation of crop physiological processes, one strategy can be priming. And priming is basically like we expose our crops to a mild stress very early in the crop cycle, and that strategy can help protect the plant against future more severe stress. And how does that happen? And I like to make an analogy as applying a vaccine to the plants. We are like providing a little, like we're exposing them to a mild stress, like very early in the crop cycle. We are not trying to reduce growth. We are just trying to trigger some stress responses at the molecular and biochemical levels in the plants. They like start producing those metabolites that are involved in stress responses that once they are stressed again in the future, they can have a faster stress response or like stronger, and they can perform better under recurrent environmental stresses. And in that way, plants can be prepared to tolerate stress conditions. And that happens through a process called stress memory. I know plants don't have brains, but and once you trigger these metabolic patterns, like this metabolic production of stress responses, they can prepare faster if that stress happens again in the future. And stress priming, there is like several different strategies. Here I'm going to focus in two that I studied in my research, water deficit priming and chemical priming. let's start with water deficit priming. So how does that work? We first need to understand that crops and their stress responses will vary according to the timing, duration, and intensity of the exposure to stress. Here I have the crop cycle of my crop model, peanut. And here, as you can see, like there are some color-coded arrows, right? So this is like when I introduce you to timing. Not every single crop stage is going to result in the same yield or plant growth reduction if the plants are exposed to stress. In this case, as you can see here, in the beginning of the crop cycle, when the seeds are like just developing and like becoming a seedling, this is a very stressful phase for the plant. So in this period, if the plants endure like even like a mild water stress, it can be detrimental to yield and can have a severe yield cost. Another phase with the similar like problem is during the flowering and pod filling, like grain filling. This also, like if the crop endures like water stress during this phase, we might have like severe yield costs. But there are other phases, like once the plant already developed and is in the vegetative stage, this is a phase that like if you trigger a mild to moderate stress, usually that's gonna result in minimal to no yield cost. So that's why like this is the physiological stage that we usually target to apply a water deficit priming. And timing is not the only important thing here. As I mentioned before, like we're talking about a mild to moderate water stress. We're not talking about a severe stress that is gonna have a huge duration that is gonna result in reduced plant growth. We're not going for that. Ideally, like we don't see like many symptoms at the physiological level. We see like very small reductions, and the majority of the impact is really at the genetic and the biochemical levels. So we really don't want like to cause visual symptoms in our plants from our priming strategy. So these three are very important here: timing, intensity, and duration of the exposure to stress. And in peanut, there is a strategy that is already being adopted by farmers that is vegetative regulated deficit irrigation. So farmers apply less irrigation and they still obtain similar yields. So this strategy like has been like studied in different like previously published research in different cultivars. And in general, like when you apply a regulated deficit irrigation in peanut during emergence to early flowering from 0 to around 45 days after planting, and then after that you restore the irrigation throughout the harvest, you can reduce irrigation up to 15% at no net cost in yield. And actually, some cultivars show some potential for improved stress tolerance. When the irrigation is reduced, they actually increase plant growth. This research focused mainly on increasing the irrigation efficiency and focus most on the environment. My strategy is now like using a similar approach since it was so beneficial for the crop, but instead of focusing on the environment, my goal here is to focus on the plant and how the physiological processes are affected by such a strategy. So in this case, I renamed the regulated deficit irrigation to water deficit priming. And in this case, my goal, besides like improving irrigation efficiency, is pre-acclimate the plants to stress. So our priming period is the same, anywhere in that window of 0 to 45 days after planting, and then we reduce the irrigation during that period. And then instead of restoring the irrigation throughout harvest, I restore the irrigation for a period so we can recover our plants and they can be compared to the non-prime plants or control plants that received full irrigation. And then I expose them to a subsequent severe water stress. And that subsequent severe stress can be within the same crop cycle or also with cross-generation stress memory, like in which the parental generation experiences this priming strategy, and we try to understand the residual effects of that exposure in the next generation of plants. So we harvest those seeds and we plant them in the next generation, and we stress the next generation of plants. And then we see like if the parental stress exposure can affect the stress responses of the offspring. So now that we cover water deficit priming, let's move on to chemical priming. And chemical priming comes from the concept that like under environmental conditions, plants are exposed to a coexistence of a wide range of stresses. And with that, like plants present some evidence of cross-tolerance and cross-priming. And in this case, like environmental stresses, they trigger like many metabolic pathways. And those metabolic pathways, like different stressors like salinity, drought, floating, some of them like share stress responses, like they share genetic pathways to trigger that stress responses. And then there is evidence that plants have enhanced tolerance to one stress triggered by the exposure to another. And chemical priming bases on this principle, but instead of giving an environmental stress like we did with water deficit priming, we give a chemical priming. We expose the plants to a biochemical clue, and that triggers these shared stress responses that like will increase like their basal levels in the plant. And once they are stressed again later, they can be again more prepared for that stress response. And then chemical priming can be applied using priming agents that are natural or synthetic compounds. Here I have three like common ones: melatonin, salicylic acid, brassinosterides. They are like basically like pheromones that the plants already produce. And those chemicals can be applied to the seeds, roots, and leaves. In my research, I decided to move on with seed priming with melatonin. And I chose melatonin because it's a multifunctional molecule in the plant. It has like is involved like in several important parts of stress responses. One of them, it has a direct action as an antioxidant and is present in different organelles within the plant. So it has like a very fast transport or efficiency to respond to a stress exposure. Has also like some biostimulator function and works like directly with the redox network. And is also like involved like as a plant growth regulator that like interacts with other plant hormones, such as auxin. And why we are applying to the seed? Because it's a cost-effective approach. And if eventually like becomes commercial, it's an easy way for farmers to have seeds with improved stress tolerance because the treatments happen out before planting. And the dose of melatonin to the seeds, like they are like very small rates. And in several crops, it has been shown improved stress tolerance and also more uniform germination and early establishment. And that's actually very interesting for my crop model, peanut, which has been reporting like to have seed quality issues due to harvest of immature seeds, problems with transport and storage. So then like with that, my research questions were, can water deficit priming with regulated deficit irrigation help peanut become more tolerant or resilient to water stress? And is there potential for chemical priming for peanut drought stress tolerance? Now moving to the next segment of my presentation, let's talk about the physiological assessment and data organization and analysis. So to conduct this research, I use an automated irrigation and whole plant lysimeter system. So I have a picture of my greenhouse at the University of Florida. So here we have 32 pots in a climate-controlled greenhouse, and each one of these pots is composed of a weighing lysimeter, a drainage container that like removes any excess from the irrigation, a pot, and a replicate like that is one single plant. And each of these pots has a soil probe, like a soil water sensor that continuously measure volumetric water content, soil temperature, and electrical conductivity. And all of those pots are sealed to prevent any evaporation from the substrate. So any differences that our weighing lysimeter records, and those measurements are recorded every 3 minutes, they're only due to whole plant water use that help us guide on this automated irrigation. And in addition to that, like every pot receives an independent irrigation that's based on the plant water use of every single plant. And what does that mean is that like this weighing lysimeter collects measurements of whole plant transpiration from the previous day, and we apply the irrigation based on that for the next day, considering like any possible like plant growths of the next day. So that's like how we applied our priming and stress treatments. And we also use like the lysimeter as an indirect indicator of plant growth. And as stress indicators, our main one was like gas exchange that we performed using the SARS-3 from PIP systems. We measured net CO2 assimilation, transpiration rate, and stomatal conductance on the second fully expanded leaf of peanuts. We also measure water potential and relative water content, but today, because of the time, we're going to just focus on the leaf gas exchange measurements. So finishing that segment, let's move on to our first case study, advancing climate smart agriculture using water deficit priming. So our objectives here were to define timing, duration, and water deficit levels for the priming treatment, and determine if water deficit priming can influence responses to a subsequent more severe water stress. And this research was conducted in two steps, as I mentioned before. We were trying to understand subsequent stress within the same generation and also residual effects that could be passed to the next generation of plants. So for the first part that we did on the same generation, we used the genotype C7616 and the cultivar Tough Runner 511. Both of them reported as moderate drought tolerant, and we had 3 irrigation treatments, control, non-primed, and primed. So here in this graph, I'm showing you the volumetric water content during the entire experiments in days after planting. So in gray here, I have the control plants. They basically just receive daily irrigation based on their water use during the entire experiment. And then I have our non-prime treatment that is shown here in blue. So they receive similar irrigation every day as the control plants up to 55 days after planting. And this is around flowering and pod filling, so a very vulnerable stage for peanut. And then at this period, they were exposed to a severe water stress. Remember, these are the non-primed plants. This is the first time that they are enduring water stress. And in here, how we expose them to stress, this is one of the multiple experiments that we run to understand the timing, duration, and intensity of priming strategies. And at this point, we already had established some previous thresholds. And every time the substrate reached those thresholds, it was triggered an irrigation. But as you can see here, differently from the control, they were receiving irrigation every 4 to 5 days, and it was less amount than the control plants. And this went on for around 14 to 15 days when the plants were having whole plant transpiration of less than 30% of the control plants. And after that, we work over the plants with daily irrigation again. Our third treatment is a primed treatment, and it's shown here in orange. So again, as I mentioned, I tested different durations and timing and intensity. And the one that I'm showing here is the most successful one that we had. And this one we were irrigating daily as well, up to 20 days after planting. After that, we reduced the irrigation to around 60% of the irrigation of the control and non-primed plants during this period. So we were irrigating with less frequency and up to another threshold that we also found from previous studies. And at 45 days after planting, we ended our priming treatment and then restored the irrigation for 10 to 12 days. And after that, they were exposed to the same water stress that our non-primed plants and then recovered. So let's move on to our results. Here I'm showing the leaf net CO2 assimilation, which was our indicator of stress levels in response to priming. So here I have net CO2 assimilation on the y-axis and days after planting on the x-axis. On top, I have the cultivar C7616, and on the bottom, we have Tough Runner 511. So ideally, what we want to see in this graph is during the water deficit priming, although we reduce the irrigation of primed compared to non-primed and control plants that were receiving full irrigation every day, we don't want to see any changes in net CO2 assimilation. We don't want to cause any significant reduction in growth, right? So this is an indicator that our priming strategy was successful because although plants were receiving less water, they were still not showing significant reduction in net CO2 assimilation. And then after this, we go to our recovery phase. And again, we don't see any difference between among control, non-primed that were receiving full irrigation and our primed plants that just started receiving full irrigation. And then also those plants are water stressed again, we can see some difference between the two cultivars that we had here. Let's get started with the C7616. So here in black, we have our control. and like they have higher net CO2 assimilation as expected because they were receiving full irrigation, right? And we have our both non-primed in blue and our prime treatments in orange right here. We didn't see like significant differences between these two treatments, but we can see here a slight trend of more pronounced reduction of net CO2 assimilation for primed compared to non-primed plants. And after like this stress was ended, when the plants were recovered, like both non-primed and primed plants were able to recover to control levels of net CO2 assimilation. When we look to tough runner, we see somewhat like different results. We can see that like our primed plants compared to our non-primed plants, they actually like were maintaining net CO2 assimilation more similar to control than non-primed, even under like very severe stress at this point in the experiment. So we can see here like a little difference. Here our plants like were becoming like somewhat more sensitive to the stress, while our tough runners, they were becoming more acclimated to the stress, like so shutting down the metabolism like somewhat later. And what does this mean is that like either strategy like can be beneficial. We really like depend on the context of the environment. Either way can be beneficial according to your environmental conditions. And so when we look at whole plant transpiration as an indicator of plant growth, we can see here whole plant mid-day transpiration and like by days after planting. And here we can see some difference in cultivar as well, like with the C7616, only showing differences between the primed and control plants only when they endured the second exposure to stress. So up to here, plant growth probably was not affected. It was just when it was exposed to stress again. However, for our tough runner 511, we already can see compared the control and non-prime plants that were receiving full irrigation, our prime plants start showing signs of reduced whole plant transpiration by mid to late priming. So probably like around here was already like having some mild to moderate like reduction in plant growth. And then like when it was exposed to stress again, it showed like what we showed in the previous slide, like somewhat an acclimation to the stress, like not as pronounced reduction of transpiration as it got stressed again. And when we look to the plant dry weight by the end of the experiment at 85 days after planting, for C7616, we didn't see a significant difference like on plant dry weight among control, non-primed, and primed plant. But we do see a trend of higher growth reduction in non-primed compared to primed plants, and some indication of improved water use efficiency as well, since those plants were like receiving full irrigation for the most part of the experiments, and they still like assimilated less carbon compared to our primed plants that like received less irrigation. However, for Tough Runner 511, we saw that like our non-primed plants, they were actually more similar to our control plants, and there was like a more significant growth reduction due to the two exposures of water stress. So this research is showing that like this is like not a one recipe fits all. We need to like really like develop a strategy that's going to be effective for the most cultivars that we can have. And at the same time, like we really need to play with those timing, duration, and intensity of the stress. But it is possible to improve water use efficiency. We just need to work with like the the genetic material and also with the environment of the crops. So the second part of this case study was like producing primed seeds to test cross-generational effects of water deficit priming. So here we use two different cultivars, COC 041, which is a drought tolerant, and New Mexico Valencia C, which is a drought susceptible. And here we had two parental plant stress exposure, non-primed and primed. And our non-primed parents, they receive like full irrigation during the entire crop cycle, like daily irrigation, and our primed parents, and after seedling establishment, we started like our priming strategy, giving around 60 to 50% of the control irrigation in like less often irrigation events, up to 45 days after planting. And from this place on, like they received like restored irrigation daily up to 155 days after planting, in which we harvest those seeds and then we plant it again. In this experiment, in which those seeds from both primed and unprimed and from both cultivars, they were planted, and both of them, the well water and water stress, received like daily irrigation up to 35, 38 days after planting, when they were exposed to water stress, to either water stress or control conditions. And remembering that like at this point, like the plants are like at early flowering, so it starts to become like a very vulnerable physiological stage for peanut. So first I would like, we didn't see like a lot of differences in stress responses for whole plant transpiration under stress, but we saw something interesting. Under early establishment, like here from 5 to 35 days after planting, we can see that like the offspring of plants that come from primed parents, they showed like higher whole plant transpiration. And that's an indication that they were accumulating more biomass during that period compared to the offspring that comes from non-primed parents. And then when we look at the results during water stress from 35 to 48 days after planting. So here I have like both of our cultivars, the drought tolerant and the drought susceptible. And on the top, I have the well water plants, and on the bottom, I have the water stress. We didn't see a significant difference in net CO2 assimilation or in transpiration rate at the leaf level. However, there were some trends of higher net CO2 assimilation under similar transpiration rates. And as a result, under water stress, we saw for both cultivars improved water use efficiency, which is basically like dividing the assimilation by the transpiration of that plant. And this is very interesting to see that like under well water conditions, we didn't see a difference, but under water stress conditions or the offspring of primed parents like showed improved water use efficiency. And that like when we look at plant dry weight, like biomass accumulation, here I have like the comparison between well water and water stress, and then for non-primed and primed of each cultivar. So here we can see like there is not a significant difference between water stress, non-primed and primed offspring, but we can see some trends of higher biomass accumulation under water stress for primed compared to non-primed offspring. And when we look at these two for our drought susceptible cultivar, we see like similar trends. And also, we see the difference between well water and water stress in each priming category, we can see also like a higher impact on plant growth from water stress in non-primed compared to primed plants of both cultivars. And when we look at the plant water use efficiency, so in this case, we are dividing like the whole plant dry weight in grams divided by the liters of water transpired during the entire duration of the experiment, which we call like cumulative transpiration. We can see that like for most results, like there is no difference between well water or water stress plants. But for our drought tolerant cultivar, it's interesting to see that under water stress conditions, there is a significant improvement on plant water use efficiency under water stress for primed compared to non-primed offspring. And these are this is the end of our water deficit priming studies. And keeping the lookout if you are interested in this research, like it's under revision, and we hope to publish this soon. And now moving on to the second case study in advancing climate smart agriculture and using seed priming with pheromones. So for this research, our objectives were to test melatonin rates for priming peanut seeds and to evaluate whether seed priming with melatonin can reduce water stress of peanut plants. And for this study, we started with like a melatonin rates experiment. This is the first time that like seed priming with melatonin is applied on peanut. So we really needed to get like right in the beginning of the research. So I did a literature review in seed priming with melatonin, and for other crops that like are from similar family or genus of peanut, there was a range from 0 to 200 micromole of melatonin reported as improving like early establishment and also stress tolerance. So that's why we tested seed priming with 0, 50, 100, 150 and 200 micromoles of melatonin. And for this study, we used two cultivars, the Forerunner 511, again, a drought-tolerant cultivar, and New Mexico Valencia A, a drought-susceptible cultivar. And then these are the results from our melatonin rate trial. And here we used radical length and germination at 7 days after planting as our indicators. And for both cultivars or more promising results, they were found for the rates of 50 micromole of melatonin. So that's like what we moved from for the water stress experiment. And then, so this experiment, again, like we used two cultivars, and our non-prime treatment was like basically like DI water treatment, and our primed was with 50 micromoles of melatonin. And then here again, like we had two irrigation treatments, a well water, like full irrigation daily during the entire experiment, or a water stress in which the plants received daily irrigation up to 40 days after planting, and then irrigation was ceased until the plants were transpiring less than 30% of the well water plants. And then let's move on to our results. Leaf net CO2 assimilation here. And then remember, like this is our drought susceptible cultivar, and this is our drought tolerant cultivar. On top, we have well water conditions, and on the bottom, we have water stress. Very interestingly, for drought susceptible, under well water conditions, we can see a higher net CO2 assimilation of primed compared to non-primed plants. And once these plants are water stressed, they actually do quite the opposite. They reduce faster net CO2 assimilation compared to non-primed plants under like extreme water stress. And for Tough Runner 511, we see some very slight trends of higher net CO2 assimilation for primed and non-primed, but we see significant results under water stress. Very like the opposite of our drought susceptible cultivar. We see the like Tough Runner plants, they were primed plants, they were able like to maintain net CO2 assimilation for longer during extreme water stress. And then another part of like this, another result is like whole plant midday transpiration. Again, we can see our primed plants here in orange, like showing like trends of like higher plant growth during early establishment and also like mid crop season. And under water stress, we can see like higher slope of reduction of transpiration, and also like that probably like those plants like were bigger when the water stress started. And in this study, we actually moved on to try to understand like what was going on in the biochemical level. So I have like this table. I don't want you to pay attention too much to all these numbers. This table, like what I want to accomplish with this is just showing a list of the antioxidants that we analyzed. That we also had the lipid peroxidation indicator to indicate like membrane damage due to water stress. We also analyzed like reactive oxygen species and protective osmolytes. So in my other goal with this table is as well to show that in our analysis of variance, all of them were significantly different between well water and water stress plants, as well as like between non-primed and primed treatments. And let me see. And for this biochemical analysis, we did in 2 time periods. One at early stress, like just a few days after irrigation was ceased, as well as like by late stress, like by the last day of stress, which was after 14 days that the irrigation was ceased. And then for the first one, we saw mostly the irrigation and priming effect. And for under late stress, we saw also an interaction between the water levels and the priming treatments. So this is very complicated to like grasp, right? So let's go to like a summary of those results. So for that, I put like all those measurements into a principal component analysis to try to understand like what is going on the biochemical level that explain like why primed plants were performing better under water stress, right? So here I have on the y-axis the principal component 2, and in the x-axis I have principal component 1. Let's not focus on the graph for now, and let's focus to the right just to know that like the biochemical variation, like 90% of that was explained by these two principal components. And the first one is basically like an orchestration of like different activities of antioxidant enzymes at both early and late stress, or at early stress, or only late stress. So that's like what is interesting to see, like how these antioxidant enzymes like interact to each other to improve the stress responses. And principal component two, it was represented by reactive oxygen species and lipid peroxidation at both early and late stress. So now let's go back to the graph. Let me go through the treatments as I show you the cluster separation. So the most obvious and like first separation I want to show you is between filled symbols and empty symbols. And this is like the water stress treatment and the well water treatment. If you see here, which like makes sense, or water stress plants, they are showing like higher levels of principal component two, which are reactive oxygen species and membrane damage, which makes sense, right? Than our well water plants. Our second separation is between drought sensitive and drought tolerant. So here's a difference in the symbol shape. Here I have diamond symbols, and here I have triangle symbols. And this makes sense as well. So our drought sensitive, they were like dragging towards more that quadrant, in which there is higher reactive oxygen species accumulation and lipid peroxidation, and actually lower of principal component one, which is like improved antioxidant activity. And our drought tolerance in the opposite is exactly that, like reduced reactive oxygen species and lipid peroxidation, probably due to improved activity of the antioxidant system. And our third like separation is between non-primed and primed plants. Very interestingly, let's start with the well water ones. So here we are moving from blue to salmon, which is like blue is represented by non-primed and salmon is represented by primed plants. And as you can see here, for well water, doesn't change so much for principal component two, but it changes a lot for principal component one. So both cultivars, they are moving towards higher basal levels of antioxidants, even under no water stress conditions. And more interestingly, when we look at the water stress plants, again, like here we are moving like our drought susceptible cultivar is moving towards with a like higher slope than our drought tolerant cultivar. We are moving towards lower levels of principal component two, which are the reactive oxygen species and lipid peroxidation, and moving towards like higher antioxidant activity. And the same is seen like, but not as pronounced for drought tolerant cultivar. And then from those results, like we also measure plant dry weight in the end of the experiment. And again, like due to some variability within the data sets, we also didn't see significant differences, but we saw like some trends of higher plant growth reduction on non-primed plants exposed to water stress compared to primed plants of our drought-susceptible cultivar. And interestingly, we saw on the well water conditions the major effects for our drought-tolerant cultivar, in which well water plants had like higher biomass accumulation. That was not significant, but like we saw those trends. And with that, we developed this diagram, like we've suggested mechanisms for improved early establishment and stress tolerance promoted by melatonin priming. And so seed priming with melatonin promoted like faster, more uniform germination in peanut. And then as those plants grow and they were under well water conditions, they had improved gas exchange, higher plant growth, and higher basal levels of antioxidant activity. And under water stress, those plants produce had more antioxidant activity. They also had higher accumulation of protective osmolytes, less production of reactive oxygen species, and less damage of membranes due to water stress. And this research is just like out there, like just got published last week. And here are the details if you want to read more about this. And with that, my concluding remarks are that water acid priming effect on the same plant generation, we saw a significant impact of the timing and duration of the priming strategy, and we saw some potential for improved stress tolerance and water use efficiency. For the water acid priming effects on the next plant generation, we saw some faster early establishment, like the plants were growing faster, like during that vegetative phase. And we also saw increased plant water use efficiency. And for the seed priming with melatonin, we saw that like 50 micromoles is like the most promising results that we can have. We have improved seed germination and early plant establishment. And we had some growth promotion under known stress conditions for a stress-tolerant cultivar and underwater stress for a drought-susceptible cultivar. And also we saw, regardless of the environmental conditions, increased antioxidant activity and lower reactive oxygen species accumulation. So with that said, I'd like to acknowledge like all everybody that was involved with the funds and the financial aspects of this research. Without them, like nothing of this would be possible. I would like to thank PP Systems for inviting me here today. And I also would like to acknowledge like all the amazing researchers that were involved in this research. Like without them also nothing of this would be developed. And thank you for joining the webinar today. And this is my information if you want to connect in any of these professional holds.
Thank you for attending the webinar. Contact us today to learn more about the CYRUS-4 portable photosynthesis system, the 4th generation portable powerhouse elevating the high-level field research experience worldwide.